I guess I'll start by introducing Ann to you. She's the president of Lilly Neuroscience at Eli Lilly and Company, and a valued partner to ADDF at Eli Lilly in our diagnostics accelerator and in other ways as well. We're trying to develop clinical trials together. Ann has over 25 years of experience in the pharmaceutical industry and brought new medicines to market, which is quite an achievement. It's been said that 90%, Judy Lewent, who was the chief financial officer for Merck for many years, was quoted in a journal I was reading at the time that 90% of the people who work in the pharmaceutical industry will spend their entire careers never working on a drug that comes to market. That's how hard it is to imagine we're going to get a little white pill someday that's going to prevent and treat, maybe cure Alzheimer's disease. It's an incredible thing to do. I wanted to just give us an overview briefly of why our partnership with Lilly is so important, and I have described the ecosystem that leads to the development of a drug. Alzheimer's really, in the modern era, wasn't discovered until about 1970 when pathologists in academia figured out that senility was not a normal part of aging. So in the history of human beings, senility was thought to be a normal part of aging until about 1970 when these pathologists looked at the brains of older people and saw that they had plaques. And that was the first step. And that wasn't so long ago if you think about it in the context of history. And then in 1984, Glenner and Wong, two scientists, discovered that these plaques that had been described by Alzheimer in 1906 contained a molecule called beta amyloid. And it was that discovery that then became the target for the development of new drugs that we are seeing on the market today, hopefully soon to market. One of the key aspects of this is that we need biomarkers. Imagine trying to develop a drug for heart disease to reduce cholesterol. If you couldn't measure cholesterol in the blood, that's a biomarker. Now we can develop drugs that lower cholesterol, and we know that doing that we can prevent heart attacks. Same thing with blood pressure, with hypertension. Imagine if we didn't have a blood pressure cuff, which we didn't have until around 1900, couldn't develop hypertension drugs. So to kind of describe the ecosystem real briefly, and it takes about 12 to 15 years and about $1 billion to develop a drug for any indication, or most indications. We started, the field kind of started, I got a phone call from somebody at the University of Pennsylvania in 1999. And he said he had a crazy idea, couldn't get any money for it. He wanted to take the same dye that Alzheimer used in 1906 to describe Alzheimer's disease in a 52-year-old woman. And he wanted to develop that dye as a PET agent so anybody could go to their radiology office, get an injection of a microdose of radio-labeled PET agent that would go into the brain, it would bind to the plaques, and it would light up on a PET scan, and we could make a definitive diagnosis of Alzheimer's. That happened in academia. Our president of research at Eli Lilly was involved in that program as a fellow in an MD PhD program. We paid his salary, and in 2005, that program was spun out into a small biotech company with about $7 or $8 million from venture capital. 5 years later, Eli Lilly bought that company for $800 million in milestone payment. And you got to wonder, why would Eli Lilly pay $800 million for a test that would definitively diagnose Alzheimer's disease? Well, what they did was they looked at the brain. They used that test to look at people who were entered into a clinical trial of a drug called solanezumab. See how many people in that trial actually had Alzheimer's disease and actually had the target of the drug at that time, which was beta amyloid. It turned out about 35% of people in the trial did not have Alzheimer's, even though they were recruited by experts, not have amyloid, which was, of course, the target of the drug being investigated. Of course, how could the clinical trial succeed under those circumstances? And so, fast forward, we have one drug on the market today. The first one, Aduhelm, came out of Eisai Biogen. It got what's called an accelerated approval. What does that mean? It means that the FDA determined that because these drugs were shown with the PET scan, to almost completely remove amyloid from the brain. So you do a PET scan before the trial starts, then 6 months later, 12 months later, 18 months later, you could see that the amyloid in the brain was completely gone, proving that the drug worked in patients who actually had the disease. So the first approval for a disease-modifying drug in the history of the world actually started with that phone call I got from an academic scientist to a philanthropy, spun out by venture capitalists, bought by big pharma, and ultimately resulting in accelerated approval. And it took 25 years to do this. So that's the kind of ecosystem that we're working in. We can't do the kind of things that big pharma can. Big pharma might not take the kind of risks that the philanthropy take. So that's why we work together in a productive way at Open Years. And what's your take on this ecosystem and the kind of talked about?
No, it was a wonderful summary. And I just want to echo what you're saying, that vision that the ADDF had to invest amyloid PETs, the way to diagnose Alzheimer's, is the reason we today. I can agree with you. We had trials that knew targeted amyloids, that if you have people in the trial who don't have that disease, you're not going to have successful trials. So it was transformative. And I will tell a story that when Dan Skowronski, who's the young scientist that you're mentioning, and he presented the first was to actually only enroll people with amyloid formation in the brain, there was actually laughter at the first scientific meeting. People like, oh, this is really not going to work. And why are you just going to take forever to enroll the trial? Now, you wouldn't do a trial without measuring amyloid in the brain. You wouldn't even consider it. So it's just funny how science evolves, but you have to be a visionary at the beginning and sometimes take ideas that sound a little crazy at the beginning and then invest in them. So I just wanted to thank the ADDF, and Dan, if Dan were here, he would say that the same is that someone having vision to take risk to advance the field forward is incredible. But this was transformative. A personal story is my mom actually, unfortunately, died from Alzheimer's disease over 10 years ago, actually almost close to 15 years now. When she was diagnosed, it was only a clinical diagnosis. There was no PET. There was no biomarkers. So you only knew for sure that she had the disease after she died. Imagine the uncertainty. Essentially, it was a diagnosis of, let's rule out everything else. And then, yes, she has Alzheimer's disease. And we always still wondered if there was something else we could have done. So transformative for the space. But what that's led to now is the success that we have, medicines, either, as you said, approved or near approval on the market that are a response to being able to do that. As you said, biomarkers they're absolutely key. And so we are with the Diagnostic Accelerator, and the fact that PET diagnostics is wonderful, but we need to take that step forward to blood biomarkers. The reason I feel so strongly about that is we're going to build on the success that we have with these results, the fact that we're able to slow the progression for people in early Alzheimer's disease. Can we move even earlier in people that have no symptoms yet and attack that plaque in their brain and potentially prevent people from ever getting Alzheimer's disease? But the fact is, you might walk around not knowing if you have that, and we can't all walk around and get a PET scan. So a blood test that you can get would be in transformative space, because the most important thing with Alzheimer's disease, we'll talk about it, I think, today, is the urgency to get an early diagnosis. That's where we can have the best impact and slow the progression the most. And that's consistent across all the medicines in this space.
Yeah, so donanemab is the not the chemical name, I guess you could say, of the drug that Lilly is hoping to bring to market soon.
Yes.
With the meeting of the FDA occurring.
Yes, on June 10th. So an update for all of you is that the FDA had asked for an advisory committee on donanemab, and we just had the data announced publicly this morning. So you get the breaking news that it's June 10th. Yep.
So Leqembi is the Eisai brand name for the Eisai Biogen drug.
Yes.
But the donanemab clinical trials that Lilly did were different in terms of dosing and the use of the scans and things like that. Could you talk about, because it really was, we'd learned how to do clinical trials on Alzheimer's today. We didn't know how. I did a clinical trial in 1981. When I look back at that, it was so crude. I mean, it was uninterpretable probably. But I think the major advance in the field in the last few years has been using biomarkers and really knowing all the metrics to do good clinical trials. So investors, for example, including pharma and others, can have confidence that at least the clinical trial will be good enough that you'll get an answer that's a reliable answer if the drug works or not. That's what we want to know, but at least we know how to go clinical trials. But Lilly did a very innovative approach to the clinical trial in terms of these things. I wonder if you could talk about that, because it's really, I think, interesting in terms of stopping rules and limited dosing and things like that.
Yep. Our commitment is to continue to innovate. And I know you're joking about those crude trials, but it's important to remember that every study, including the one that I'm sure you did, you had insights that you applied to the next trial. So that's what we did at Lilly, is every time we had an answer, sometimes the answer was not a positive one, but as we looked at how the patients progressed and which patients responded, you took that insight and applied it to the next trial. So some people ask me, is it a surprise that you had success? It was not all a surprise. You could see the trajectory we were on with every insight that we had. So one of those insights was, obviously what these medicines do is they target amyloid plaque. What it does is it essentially sticks to that amyloid plaque in your brain, and then it stimulates your immune system to clear the plaque out. So that's the goal of this. It's not unlike cancer, where you try to, get the tumors out of the body, clear the tumors, and then hopefully they don't come back. And so that's the same goal here. And so we felt very strongly, just like in cancer treatment, once you clear the tumor, you don't need to keep putting patients through going to the doctor and getting infusions. People want to live their lives and move on. And so we looked at in our phase 2 study and had people that stopped dosing once they cleared their plaques. And we followed them. And then we did an even bigger trial with the phase 3 trial to say, does this work? And what we saw is when people stopped their dosing, they continued to respond. In fact, they continued to separate even more from placebo than in the trial. So what that told us was that we're onto something here, that once you clear that target that you're after, just like in cancer treatment, you can stop. Now, and then people go back to essentially a natural accumulation of amyloid, which is very, very low. Remember, this disease takes 20 or more years before you start to see symptoms. Unfortunately, it's a disease that just starts, sometimes decades before you start to see the symptoms. So we felt very strongly that you don't need to keep dosing patients, and it played out in the trial. Another important area, you mentioned biomarkers. So one other area that we wanted to confirm was tau. You've heard us talk about tau. The way tau plays a role in this disease is that you have amyloid, and that enough amyloid pathology can lead to tau. And tau tangles in your brain are what actually drive most of the symptoms that you experience with Alzheimer's disease. So what we wanted to test was, different levels of tau, does that help determine how you respond to these medicines? If you have more tau, our theory, what I'm going into, you always have a hypothesis in these scientific experiments, is that more tau will mean that it's harder to respond, less tau. You might respond better to these medicines. And we saw that. And you know the results well, Howard, that the earlier in the disease, the less tau you have, the more slowing of this disease you'll have, the less risk that you'll progress into the next stage. Really important factor is, can I reduce the risk that I move to moderate Alzheimer's? And so we were able to show that, yes, with less tau, we were able to respond much better. Now, people that still had quite a bit of tau in their brain, they still did fine. They still responded. But the really dramatic numbers were as you went earlier. We want to do that particularly to drive some sense of urgency here, because probably the thing that frustrates me the most is when people tell me, I went into my doctor because I was having cognitive symptoms, and they said, well, let's see if you're still having these symptoms 6 months from now. Do you think if you had like a really persistent cough, your doctor would say, well, let's come back in 6 months and see if you still have this really concerning cough that could be lung cancer? You would never do that. And so we need to have doctors and patients and others feel the sense of urgency. Something's not right, as you said. There's senility is not something that you should expect with aging. And so giving people that confidence to advocate. And this results showing that the earlier in disease, the less tau you have, you can respond even better, is incredibly.
And just to be clear, the tau is in neurons, in brain cells.
Yes.
And so when you see a lot of tau, it means that a lot of neurons are dying and degenerating and not functioning well.
Yep.
And there was a long story about how many trials were failing because we weren't treating early enough.
Yes.
And in fact, that was true because we were treating mild to moderate dementia patients, and now we're treating early Alzheimer's disease. And the way I thought it is very innovative because, again, it was the PET scan, because now there's a tau PET scan. And we know from pathology that the disease generally, but not always, marches through the brain in a very ordered way. starting in the memory centers and then going. And so you can map the tangles at autopsy and see how there's these what we call BRAC stages of the disease that, add to the clinical staging that we have. With the tau imaging, you see the same marching through the brain of tau, and we can treat people early, not just based on their symptoms, like mild cognitive impairment, which is a stage of the disease before mild dementia, but we can use the tau scanning. I guess the question for the company and for the practice of medicine is, will everyone be required to get a PET amyloid scan before they can get on these drugs, because they do cost money? What will be the role of tau imaging in determining who should get the drug? Will blood tests, so right now the PET scan went in my practice in Manhattan, and generally the PET scan is about $8,000, and it wasn't covered by Medicare until recently. And I think the reason probably is because they wanted to make sure that the right people were getting the drug, and we weren't paying for these drugs, which are actually not more expensive than cancer drugs or biologic for multiple sclerosis or any of these. The average wholesale price is well within range of that. But to make sure that the people, just like in the failed clinical trials, the people who are getting the drug are people that actually have amyloid in their brain.
Yes.
And then we can stage them, because we want to make sure that we're catching it early enough, but not too early. It's a Goldilocks strategy, not too early, not too late. And so the blood test, I know Lilly has just announced a partnership with Roche Diagnostics for a blood test that people are asking about, and LabCorp and Quest are offering now. We're not sure what technology they're going to be ultimately using, and whether it'll be distributed through Roche's 70,000 placements or through other tests like C2N. But the p-tau217 test, maybe you could talk, it's a blood test. It'll probably cost a few $100s. It'll be in the work stream of average doctor. You go into the doctor's office, and this is a little bit facetious, but people will go into the doctor's office, presumably get maybe a brief cognitive test. They have some impairment, a little bit of impairment. The doctor can order a blood test. Imagine a blood test for Alzheimer's. It's here. Yeah, it's here. We're just kind of disseminating it, validating it, rolling it out into the various states. Yes. The C2N test is now available in 49 states. The only state it's not available in is New York, because we have the most stringent rules and qualifiers for doing those tests. So I don't know, maybe I told the whole story there.
Yeah, no, I'd love to comment. No, I share your passion for biomarkers, clearly, from my own personal experience and then from the success of the trials. Maybe to start with your comments around amyloid diagnosis, because some people might be sitting there saying, well, what is the best way to test if I have amyloid? And the good news is there's a number of choices. So there is the amyloid PET scan, and I would say that's still, certainly in clinical research, kind of the gold standard. But as you said, the accuracy of blood tests now is quite impressive. And one of the reasons we got into the space was we wanted to make sure we set a high bar for science, the kind of bar that I know the ADDF sets as well. And some people were having, the tests out there had different levels of correlation with amyloid PET, because new tests should always correlate with the gold standard that's out there. And so we wanted to make sure that we showed, and I know you're passionate about this as well, that there was good correlation with the other biomarkers out there. So we entered into that blood test space. Our goal with any tests really is to raise all tides. So we're not a diagnostics company, we're a medicines company. So our goal is to make sure that all good diagnostics go out there. So we partner with Roche, we partner with Quanterix, we partner with many, certainly the ADDF was our first partner, to make sure that more diagnostics are out there. There's PET amyloid, there is blood diagnostics, and then also there's actually lumbar puncture, which doesn't sound fantastic, but is an option that many physicians still like to use. That you can also test if you have amyloid. So there's a number of different ways. And the FDA actually in the labels on these medicines, both current and the future, will just say confirmation of amyloid. They won't necessarily say which way that you have to do it. So it's a little bit of a doctor preference. You mentioned, though, about tau PETs. So what we found when we did the tau PETs was that people across the spectrum of tau responded. So we don't believe that you need to get a tau PET to get diagnosed with Alzheimer's disease. The fact is that people respond to these medicines across the gamut. In fact, we actually had another study that people had no tau in it, and we looked at the biomarkers, like did they clear their amyloid, and the results were even better than in people that had tau as far as that biomarker clearance. And so no, just as you're sitting there thinking of all these tests, we don't believe that tau vid or a tau PET scan is required to diagnose Alzheimer's disease. It can only be used if you want to understand like how advanced it might be, but we believe that people across the board will respond. And maybe just one other thing that I know you're very passionate about is because we saw that response where people were clearing amyloid plaques in that earliest stage so well, we actually have another trial that, as I said, is about actually catching people that have very low levels of amyloid and no symptoms and seeing if they can clear that plaque altogether. We actually enter people into that study with the blood test.
And that's a prevention trial to prevent symptoms in the first place, which is really another great outcome of this whole thing. We talked a little bit, you and I, about clinical meaningfulness and how I speak to my patients to convey to them whether this is worth taking. In the fact, that's what they want to know, the side effects and that kind of thing. So I think in terms of, let's say it's an average of 30% slowing decline over placebo, what does that mean, and how do we convey to patients the benefit and, of course, the risk, which are maybe really serious in only 1% to 2% of patients, so kind of safer than most cancer drugs. But how do we convey to patients the value of this new category of drugs, the monoclonal antibodies, the amyloid?
Yes. Yep. So as Howard's mentioning, what we saw in our trial is you saw like a 35% slowing of the decline that you would have had. But you're right. In an individual patient, you don't have a control, right? So how do you say that that's different? So we introduced, actually, I came from the cancer space. So, and Dan has a lot of cancer experience as well. And so we actually took some cancer concepts and applied them to Alzheimer's disease. This is also first in the field, which is, can we reduce the risk that you progress to the next stage? So if any of you have been involved in cancer trials, that's a big part of cancer trials is, can you reduce the risk of metastases? Can you reduce the risk that you move to the next stage of cancer? So we essentially, first time we've done this, we applied that philosophy. Can you reduce the risk that you move to the next stage? And there's actually not that many stages of Alzheimer's. There's MCI, mild, moderate, severe, and unfortunately death. So those are the stages of Alzheimer's disease. So patients in the trial were either MCI or mild. So could we keep them from progressing to mild and moderate? And the fact is that we reduce the risk that you progress to the next stage by 40%. So that's the kind of result. So hopefully that's maybe more easy to internalize than what 35% slowing means is we reduce the risk that you didn't progress of progressing to the next stage. Probably the one, though, I'll tell you that's most meaningful to me is, could you give people time to stay where they're at? Like a lot of people, especially with early Alzheimer's, they say to me, if I could just stay where I'm at, I'm able to still do the things that I love. For my mom, it would have been taking care of her grandchildren. One of when she's here today, that was the love of her life, and in her early days, she was able to watch her grandchildren on her own. If you had given her more time to do that, that would have meant everything to her. And so we institute that measure, which is how many, what percentage of people stayed exactly where they were at clinically, functionally, and almost 50% at 1 year stayed where they were at. That was just one measure that we instituted in the trial. Many of them longer than that, but you have to define kind of at the beginning what things you're going to measure. And so at 1 year, nearly 50% of people were where they were at. And so to me, that was actually, that was probably the one that got tears in my eyes, because if you could have told me on one part of your taking your grandchildren and creating memories that staying them down throughout their lives, I'm about right telling them.
There's one more thing. So even at 50%, there's another 50% that are into, and if you take the 35%, then there's 65%. So, past question, what's next? And we know, and IC plays a long history, so the labs bank. There's been a big sea change in the clinical development space where 5 years ago and more, 65-70% of the drugs were anti-amyloid, like tau drugs. Now 65-70% of the drugs in development are non-amyloid, non-tau, anti-inflammatories, and multiple other targets. Our scientific strategy is recognizing that aging is the main risk factor for Alzheimer's disease, and we know a lot of mechanisms of aging that we're trying to apply to discover those hemoglobins, because we want to get to 100% slowing. And so we're looking at an era where we're going to need new biomarkers like that. Groups patients have inflammation because they really don't progress that, in a PET engine, blood testing, on and on. That's why we're involved in this $100 million program that you're involved in. with Bill Gates and Jeff Bezos to develop these new biomarkers to identify these other pathways to get from that 65, 70% or whatever that anti-amyloid doesn't get at. And then ultimately envision that these precision biomarkers lead to combinations therapy. And all the challenges that you can learn from the cancer space as far as how to conduct these combination therapy trials, because they're quite complicated. So I wondered if you want to comment on the Lilly pipeline. Sure. And, as we look to the future, what's next?
Yes. Well, maybe, and to go back to your comments about the 50% slowing, just to give people reassurance, by the way, I'm a sinner. Well, that was didn't measure people that stay where they're at. There are still other people that still responded very well, in addition to those that didn't progress at the same rate it would have. But it was one measure to say how you could stay exactly right what everybody excited about. But you're absolutely right. Just like cancer, there are going to be multiple ways with type of system to real that amyloid gene apology once you can get the symptoms Alzheimer's disease and the methods to tau. So we have medicines for additional medicines in the pipeline that we are developing for amyloid targeting, but also for tau targeting. And we think that at some point those go things together, particularly as people, as I said, then have the symptoms. But then even on something is what we still want to do and what success will to do is build on that success to go earlier, just like in the cancer. A lot of time you see a medicine is launched for metastatic cancer, but then eventually it ends up in the adjuvant setting and prevents people from ever looking even at a metastatic setting. The same concept that here as well is how can we go, and I know ADDF is very passionate about this, to drive brain color such that people never adopt the pathology of this disease. And inflammation in the brain is one of those main things. And so we're fortunate that we don't just have an Alzheimer's portfolio. We have a diabetes and obesity portfolio at other places, immunology as well. And so we think that some of those medicines can help in inflammation and some of the cascade that causes dementia later in life, which you can control and stay healthy and we're not just your brain, but the rest of your body earlier in life. It reduces your risk of getting Alzheimer's or other types of dementia. So we'll be able to stay in trials, types of dementia, and particularly in prevention, because that's our passion is to move earlier currently in this disease.
Yeah.
And then you and I were talking before the start about actually working in the genetic medicine where you actually change people's risk factors. Right. So that's another level of innovation that's coming in the future. So it's real, and I think it's going to happen.
Yeah, I mean, we're at a successful career in society and living. And now you find yourself heading up the commercial side in Alzheimer's. That was 2 years ago?
Almost 3 now.
Yeah, almost 3. Right? Yep. What was your motivation and what is your goal?
Yes. Well, I think as you heard me mention, and like many of you, I was personally impacted by Alzheimer's disease, and so lost my home, and as I said, was a few years ago. And one of the things I'm most proud about at Lilly is that we never made Alzheimer's disease, and similar to the ADDF, calling the two of us have been some of the longest in this space. But we've spent now 5 years. We added up the other day $8 million that Lilly's alone spends. I know there's been much more spent probably in this space. And yes, to really get a return on that investment, you have to have vision for that to keep going. But I know our CEO at the time said that we don't give up its heart. That's when we have to dull down. And so we never gave up on Alzheimer's disease. Is it just about 3 years ago when, result in the phase 2 study, we decided to double down and Lilly go all in on investing in additional trials, expanding to the phase 3, etc. And the CDO approached me, and I immediately responded saying, yes, this is something I'd love to do. It's because Tamara, the heck, I know she's looking down the heaven and saying, look at what you've done. And she was such an advocate of women in science. My sisters and I are all in the scientific fields and in our areas. And I hear stories from her friends, and she would write about her $3. So I think she could tell people that I was working all the time to see is that how it can be great girl or along.
The difference between the two proteins, both in the chemical structure and the pathological effect, and secondly, are there other of the demonic proteins out there that can cause Alzheimer's?
So the pathology of Alzheimer's was where research started because you could see what was wrong. And now we're talking more about the biology and what's happening. But in different proteins, the thing they have in common is that they have to undergo a process called misfolding. So a protein is made by the DNA and has a sequence. And then because of that sequence, it folds into a certain three-dimensional conformation that allows it to be functioning in biology. When proteins misfold, they become a toxin. Now, through evolution, every cell in our body manages misfolded proteins because it's happening every minute of every day in every cell in our body because of oxidation and just being on Earth. So tau is in the neurons. It plays a certain role in the way synapses communicate and so on. Amyloid is, we don't really know exactly what amyloid is today exactly, but it's also in the neurons and gets deposited when the process in the cell for getting rid of misfolded proteins gets overwhelmed, which happens with aging, because with aging, the process, this garment disposal system on the cell, they get rid of the misfolded protein starts to get overwhelmed. The cells die, they spin out into the outside the cells into these plaques, and that's how the plaques form. The thing is, there's not just two misfolded proteins in the brain, it's probably hundreds, but it's just the up and blind man seeing the elephant. We're seeing amyloid, we're seeing tau, that's where Alzheimer's is like. There's also other molecules that misfold, like Parkinson's disease and Lewy body dementia called alpha-synuclein. There's TDP-43, which happens in a form of dementia called hippocampal sclerosis. And so we're realizing now that Alzheimer's disease is not just, especially indications, not just two misfolded proteins that result in plaques and tangles, but many misfolded proteins. And that may be why the strategy to have monoclonal antibodies is only 30% effective. I'm roughing it out here, because we're not getting at all in misfolded proteins. 40-50% of people with Alzheimer's disease also have misfolded alpha-synuclein in their brain. So we're not seeing that. So it's gotten much more complicated than we thought. But by having drugs that attack this process in itself, this called autophagy, we might be able to restate equilibrium and homeostasis for all the misfolded proteins that are happening with aging in every organ, and particularly in the brain.
Good morning. Thank you very much. So you guys were speaking about targeting the plaques, so activating the immune system to help with the clearing of the plaques. And you sort of answered this a little bit in the last question, but for all of us looking to prevent it, and the blood marker testing, that's amazing. What are some things people are doing to prevent the reoccurrence of the plaques after they're eliminated, and things that we can do for the prevention, if possible?
So when you have what happened, one of our board members from Oslo has, from Helsinki, did the first randomized clinical trial of prevention for Alzheimer's disease and actually cognitive aging. So what she did was she took exercise, diet, managing diabetes and hypertension, socialization, remaining engaged, things like that. And she took one group where they had to come in at least once a week and make sure that they were implementing what they were supposed to be doing. And she took another group, which was kind of the placebo, and just taught them this educational material, but then just let them go. So they weren't necessarily compliant. So they had it's a randomized clinical trial of prevention. And it was the first time that this happened. And she showed that these lifestyle interventions, say switching for heart disease, also work to slow down the progression like the common hearing, aging, or Alzheimer's. That study is being replicated now in 62 countries around the world. But in addition, you were doing what's called the finger trial that's finished geriatrics in 2.0, where we're adding the leading anti-aging for it, which is metformin, to the lifestyle intervention, which as you'd add a stack and to the lifestyle intervention to prevent heart disease. And the point is that we really had amazing advances in prevention in the last 10 or 15 years, so that there is data that the onset of clinical symptoms of Alzheimer's can be delayed. And it's been modeled that if we can delay the onset of symptoms from Alzheimer's and related dementias by 5 years, we would reduce the number of cases by 50%. And most of us think that's an achievable goal.
Thank you. Building on the question of preventative care, could you share your thoughts on the diabetes drugs that there seems to be some benefits emerging from their disease with anti-inflammatory. Yes, I'm okay.
Yes, very true. So obviously, just to echo what was just said, what's clear is that diet, exercise, being at a healthy weight, all of those translate into benefits when we're older. We all know that in other ways, heart health, cancer health. But what's emerging now is also the data that side, and that helps with brain health as well. And so, we actually have a very large diabetes obesity portfolio. And so we're definitely looking at large trials that we already start looking at, kind of mine the data there, that there have been reports either with Lilly medicines or others on this impact on brain health. So we will be exploring that. I do think, and that's very much in that preventative space. We drive greater, human health so that some of the diseases that impact because of obesity, dieting, exercise, can be prevented. So yes. So I think Lily's probably make good all by indulging that as both a leader in the most sense space material and diabetes space. So more to come on that space, but there's definitely a lot of versatile of these medicines that we have now for diabetes and obesity. Diabetes as well. Controlled diabetes is a important part of brain health, is to make sure that you are diabetic, you control that diabetes, that would mean to play your brain health as well. So we're very hopeful. Yep. The disease was impact within an alt trajectory, not just in cardiovascular health in other obvious ways, but in these more solid ways.
Could you comment on when we all have genetic testing and we're told by 23andMe or whatever that, we're not susceptible or you are? What is that testing for exactly, and what are they missing?
Well, just at the most basic level is that what ApoE4 testing, whether you have ApoE4, 3, or 2, or what combination that you have, just changes your risk factors. So just to reassure you that if you are an ApoE4 homozygote, means you have ApoE4, that does not mean you're going to get Alzheimer's disease, but it does increase your risk fairly dramatically. So that's a choice people didn't make now to test for that. We do look at the labels of the approved medicines that we expect across the field. The FDA is probably going to encourage people to get testing to see if you're ApoE4 or if you're not, or because your risk of getting some of the side effects that do happen with these medicines are higher in some of those people, but they still get benefit from these. We get medicines as well. So I do think it's an important part, and my expectation is more from that testing will be done because it informs the decisions that you can make about new studies.
Diet clean out. Or other back.
Yes. It's not guaranteed. It's not a automatic issue among Alzheimer's disease. There are other genetic markers where that is that you
Well, if we read the front page of the New York Times, they can talk about this issue of the ApoE4. So if you have double 4, you're about 15 times more likely to get it. You'll get it 10 years earlier than the average person who might get it at 75. You get it at 65. If I see a patient in my practice that their onset was 62 years old and dad had it and grandmom had, then the patient wants to know, why is this in my family and what are my risks? And also, because I mentioned, are they going to be qualified for what kind of risk will they be taking by going into taking these drugs to remove amyloid because they have a higher incidence of side effect and more serious. What we're doing, we've known about ApoE4 as a risk factor for 35 years. But we haven't been able to drug it. And it kind of illustrates how hard it is to go from the basic science discovery to ultimately getting a drug because ApoE does so many different things in the cell. So we took a gene therapy approach, spun out a company out of Weill Cornell, because ApoE2 is protected. ApoE4 is a bad risk factor. So what this group is doing, they're taking a virus, which is a non-infectious virus, putting the gene for ApoE2 into the virus. ApoE2 is protective. If somebody's ApoE2/4, they don't have increased risk because the 2 offsets 4 this. So putting this ApoE2 gene into the virus, injecting the gene into the brain of human beings, this has already been done, it's FDA approved, in a phase 1 safety trial, it proved that it was safe, and in the spinal fluid, they showed that the virus worked, the gene therapy worked, to produce ApoE2 in patients who are homozygote, they have ApoE4/4. So the next step is, to run the clinical trial in a phase 2 setting to see if this actually slows down the progression of Alzheimer's, and it would be a gene therapy approach to a very complex problem.
I have two somewhat related questions. When you're talking about Alzheimer's, are you including all forms of dementia in that terminology? And then do the tests that you mentioned for Alzheimer's, do they identify specific forms of other type of dementia? And are the treatments the same depending on what form of dementia is present?
It's a really good question, and sometimes people do tend to use them interchangeably, but Alzheimer's is a subset of dementia. So there are other reasons that you might suffer from dementia. You might have had strokes, this whole strokes, vascular dementia, other causes of dementia. So Alzheimer's is a specific one. And that's what's so important in the biomarkers to see any test for amyloid. So amyloid is specific to Alzheimer's disease. Kara, do you want to ask?
So, it seems to me after 40 years, 35 years in doing this, the people still ask me what's the difference between dementia and Alzheimer's. It's so hard to educate people about this disease. So the way I like to describe it is, imagine you came into my office and you were feeling weak and you were a little pale, and I did a blood test and it turned out your hemoglobin and your blood count was low. I said you had anemia. That's really not a diagnosis, it's describing. The next day I'd be and say, gee, I wonder why you have anemia. Do you have colon cancer and you're losing blood? Do you have sickle cell anemia, thalassemia? Are you vitamin deficient? So I'd want to find the cause of your anemia. So ego dementia is sort of the equivalent of anemia. It's a syndrome. It's not a disease per se. It's a syndrome where people have cognitive impairment that's so severe enough to cause functional impairment. It's not caused by any medication like delirium. It's a chronic thing. And then the next thing that the doctor, this is real brief, but the next thing that the doctor would do in the algorithm of assessing the patient is, first of all, do they have dementia? We can do that at brief cognitive testing in a primary care setting. And then if they have dementia, well, what's the cause of dementia? About 70% of the time it's going to be Alzheimer's disease. But it might be also, if they have Parkinson's disease, they might have a form of dementia, if they have multiple strokes. If they have a lot of vascular disease, it could be vascular. And so, historically, the value of that was a little bit limited, except as we went to risk factors. But now that we have drugs that treat the Alzheimer's form of dementia, which is the most common form by far, that it's important to have a definitive diagnosis.
And MIT gave the results of a study about 2 years ago where they looked at something other than what people had been studying. They gave some meds to mice and they reversed Alzheimer's in mice. Do you know anything? Is there any research?
That's a really big question. I'm sorry. I don't know that we can answer that. I can say that we've cured mice about 700 times.
No, that don't I know. I think it's pretty lucky mine.
Yeah. No, but to do is, you genetically alter the mice so they have the Alzheimer's gene, they had the amyloid gene, and the tau gene, and then there's literally hundreds of these different models, and then you treat them. The value of the models in drug development is to see if your mechanism works. It's not just see if you can really cure the mice. You just want to see if you have a particular drug that works as a particular mechanism. You want to test it in mice, and we're told an in vivo setting, because that's the next step in bringing the drug into the clinic for testing in patients.
Hello. My question is going back to different forms of dementia. Unfortunately, I have a younger brother who's been diagnosed with frontotemporal dementia. Are you aware of any studies specifically calling in on that?
We had a partnership with the Association with Frontotemporal Dementia, which is a drug development partnership. And we've got about 10 clinical trials for actually either have run or currently running for frontotemporal. For example, 10 years ago, we wouldn't have thought that information played any role in FTMT. Now we recognize that it does. So we're investing in companies that have molecules that can be used. Normally they try to go after Alzheimer's. It's a much bigger market, but by our investing in these companies to promote their TD program, they're willing to use their drugs and anti-inflammatories to see if they can slow down FTP by reducing inflammation. That's just one example.
We actually just see also some very medicines in development, really in FTD as well. So if you're ever interested in clinical trials, it's like all companies' trials are listed called clinical trials step out. And so feel free to keep checking there for trials. You can search on FTD and not just Lilly's trials, that anyone's there that's running trials in those states that encourage probably one of the most encouraging things I'll say is, success in medicine's ratio work. It drives investment in the space, not in the Lilly else and the smaller biotechs other places. Probably encourages investment in places like ADDF. It takes incredible risk on early phase molecules, many of which is some of these hard to treat diseases. So keep too, because I do think that there can be more medicines in the neuroscience space and the success we now had in Alzheimer's disease, inheritances, and ALS, and FTD, some of the hardest to treat diseases, but insights here and the investment and success here will drive solutions to those spaces. So check that for trial. So we'll have some.
Howard, go out 5 years from there, right? Here we are sitting here, but let's go out to 2030, right? We're sitting here in 2024, and these blood tests are coming every day. The monoclonal antibody treatments are coming every day. How will the landscape look when we get to 2030?
I've been taking care of people with Alzheimer's for 45 years, and there's been just tremendous nihilism. A lot of the consultations I get, the patients are referred to me, they say, well, which of the neurologists, they did some paper and pencil thing. They said I had Alzheimer's, and they told me to go home. That's not where we should be. I mean, there are drugs on market since 1995. There's a lot of care management issues that families need to learn about, counseling, education. What does this look like? People have, I can spend an hour and a half with a family, but under typical Medicare and so on, the average visit is about 11 minutes. So they're not getting the kind of care that they need. And I think the advent of disease-modifying drugs on companies like Lilly and the biomarkers are just going to completely change that nihilism, because for the first time, we're going to have definitive, weighted, and easy, the whole part of the work, still a physician practice, cool blood test, make a definitive diagnosis, and then have treatment options available. And this is just the beginning of a new era. It's so exciting that I think we're going to see dissipation of that treatment nihilism. And I hope with the advent of treatments still being also spin-offs on care management, because a lot of times retreatments are combined with care management to take care of the patient as a whole and the family, rather than just, go for your confusion and we'll see you later. But I think it's really going to change, and it's going to create tremendous hope. And the last thing I would say is I do think prevention is possible.
And the secret of trail is now steady remotely. Now, I've got only good news after this. Good news is our wonderful speakers are here. If you want to come up and chat with them, please do. Also, when you go outside those doors, there's a wonderful step there to get your picture taken. There's the Carolina Herrera pop-up shop. Be sure to go by that. And there was the Aaron fragrance card. Plus, there's a raffle for a live puppy. You don't want to miss that. So, thank you. The speakers will stay here for a few minutes.